Uh, Welcome to another session of RSG-Türkiye webinar series. So today's presenter is Dr. Papaleo, Elena Papaleo. Here is our social media accounts. If you would like to follow us on different social media platforms, and we would like to work with new friends. And if you would like to join us, we welcome you. Today's presenter is Dr. Elena Papaleo. The topic of the presentation is a structure-based framework to investigate variant effects in cancer and other diseases. She is from University of Copenhagen and Technical University of Denmark, and continuing her research in a member of Danish Cancer Society. Now the stage is yours, dear Doctor.
Thank you very much for the nice introduction, and it's really a pleasure to present my group and my work to all of you. I don't think I need to introduce the affiliation again, but I just wanted to say that we work as a united group despite our two affiliation at the institute and at the university. We have an approach in our research that moved more and more towards data-driven science, even if we still engage in a lot of focus study on mechanistic aspects of certain protein. And this workflow, I want to give an idea of how we work in our group. We try to take advantage of the great amount of genomic and omics data available in a publicly available repository, but also data we often get from clinical cohorts, especially in a Nordic setting. And the idea is to use a system biology-oriented approaches to dig into the complexity of this signature in cancer and other diseases. But then our really core expertise, and also what it's really exciting for us in terms of research, is to use a method that are structure-based in the field of structural bioinformatics or molecular modeling and simulation, to try to tackle the challenge of understanding the impact of mutation on the protein products. And we also have a smaller part of the group that is doing experimental validation of some of the most exciting prediction we do. We also team up with many groups in experimental cancer cellular biology to try to increase also the possibility of experimental validation and investigation. And we are starting now also to take an angle more toward drug-target interaction. Everything started for us with the realization of the large amount of data that nowadays we can get from many sequencing techniques with good coverage, with good data quality, that inform us on the genetic variation in health and disease. With this, many challenges come, especially the one of an explosion of variants that are belonging to the class of VUS, variants of uncertain significance and conflicting evidence. So those variants for which, we can see them occurring in some patients, in some disease, but we don't know the effect, we don't know the mechanism. And even in those cases in which we can predict a possible pathogenic or benign effect, or we can even have some more direct indication from experiments or from clinical data of pathogenic and benign impact, still there is a lot of unknown in terms of mechanistic insight. So what these variants are actually doing, what is the mechanism that, or the group of mechanisms that are disrupted at the structural level and for the way the protein need to work in the cell. And this knowledge is important not only for us in terms of basic research, but also with an eye and an angle on application. To have this kind of information can help disease genetic counseling and the field of disease predisposition. It can help us to try to predict before ahead variants that could trigger drug resistance. It can help to design strategy to counteract the effect of the variants. For example, if I get to know that some mutation will affect the protein stability of the protein, for example, P53 is the best example for it, I could design a molecule that try to rescue that stability and still have a functional product. It can also, along the way, and this is more ambitious, we believe that to get this kind of structural understanding of how some property of the protein are altered, together with the clinical data, other metadata on the patient, and phenotypic data or pathway-based data, could inform and be used to develop machine learning models toward predicting outcome of the patient in a more successful way. When we started to work in the group a little bit more on this and try to make a resource available that it's as comprehensive as possible, two of the PhD students at the time in the group, Mona and Christine, they also did a survey from literature that was useful for us to inform us on which tools are available, how we should look at them, and which criteria of inclusion we should have for our larger framework for variant assessment. And also to understand where are the gap of knowledge and how we can contribute to those. And this picture is just to, if you are interested in this field, you can go and read the review article. It's just to try to convey the diversity of tools that are available out there based on different methodology of different principles. Some of them are also maybe focusing on specific aspect of a protein, for example, stability or binding, and some other are maybe more comprehensive framework. I think the most interesting part of this review article is actually in the supplementary material, because the two students, they took the challenge of try to do an assessment of the usability of this tool. So they set up an experiment in which they built a Docker container, and then they give themselves a time, like around 1 hour, in which they try to assess if they can install the tool, and if this fail, for example, if it's due to a lot of dependencies, because this will instruct you also on the accessibility of the tool if you want to include this in a larger framework or protocol. And if they can pass this two-step, then there are example and test data. Can we run them? Does it run? Give results, and then we report what we find. And in this search, what we realize is that little bit of a bummer. Some of the tools actually were not even given a source code, even nowadays, that we should obey to the FAIR principle in research. But for the one, at least, that were available, we discovered that in majority of the cases, we were actually able to install the tool. That's pretty good because it's the first step. But then, unfortunately, a lot of them couldn't run successfully in our hands, even with the test case. And of course, this called for a little bit of more digging. But it's also a message for us to try, as a community of people doing software development in bioinformatics, to try to do a better job in this sense and in the direction of other people using our code. Then I want also to start to go more into what we actually do and have been building in the last few years. Our focus at the moment is very protein-centered, but with the big new discovery in the field, I think we could try to envision in the future to move this framework, also looking into variation already in the DNA structure or, for example, RNA structure. But for now, it's protein-centered, meaning we can assess mutations that are affecting the coding region of a gene and have ultimately an impact on the protein structure and then on the function. When we started to develop the framework, we started to think, okay, how this should work. We want first to understand what protein functionality can be altered by mutation, and how this eventually correlate with some experimental assay we can do for validation, so that our assessment can also instruct people doing experiment on the best readout, on the best experimental setting to pursue for the mutation. And then we narrow down in different classes that we could think about. So we want to have in our framework methodology that can cover a problem like structural stability, changes in the native folded structure that could ultimately cause misfolding, that can turn from the experimental point of view. For example, you could measure tendency to aggregation, or you could observe a higher degradation rate of the protein through the proteasome. So altered turnover of the protein. It could be an alteration. Protein are not alone. They need to interact with other biomolecules, other protein, and not only protein. They need to interact with DNA sometimes, membrane, cofactor, metals. So to have also a way to treat the effect on this interaction, at least at the local level. And this, of course, experimentally, then one could do assay, this different kind of assay of protein-protein interaction or other interaction. We need to remember that protein have an extra layer of complexity in terms of regulation by chemical modification of amino acid that are the post-translational modification. So we want also in our framework to be capable of disentangling the interplay between the PTM and the mutation itself. And we started to do it as a proof of principle on phosphorylation, and there is still a lot to do in this field for other less studied modification. We need to keep into account that some protein are enzymes, so they have a catalytic activity, and then a mutation can alter that. And we also need to always keep in account the fact that some variants, some mutation could trigger effect over long distances in the protein structure that are at the core of allostery and structural communication that are more difficult to catch. So with this in mind, then we asked ourselves, can we set up a system to use method for each of these property and try to give an atlas of effect of mutation, taking a variance of the possible mechanisms that are triggered, that we call a mechanicistic indicator in our framework. And we want to do this in a way that is standardized, that can be reproducible, that it's well curated, because we want to ultimately deliver the data to the community, not only use them for our own research. We also need to try to design a strategy that allow us to work in a high throughput manner, because we want to understand, at least with some of the approaches, as many variants as possible for as many proteins as possible. And then we need the two classes of method. From one point of view, and this is not necessarily structure-based, we want to have good methodology that can serve as an indicator for a potential pathogenicity of the mutation. And then if this can be narrowed down to loss or gain of function, because especially in cancer, these two concepts are really important for tumor suppressor and oncogene behavior. Then we want to be able to link a prediction on pathogenicity and a loss and gain of function with a mechanism. And it's where the structure-based method come even more into play, in a way that we can have a quantifiable score of properties. So we like a lot the concept of using free energy changes in different contexts. Can be the definition of free energy changes in folding, unfolding, binding, or allosteric effects. And we went very crazy in developing the toolkit and the workflow. Sometimes we had to develop our own method because we couldn't really be fully convinced of using methods from published work. And this is just to convey the message that we had been doing a lot of work, and we still do, on building up the building blocks for our resource. And they can also be used independently by other people if they have a more narrow scope than what we have. And this is done thanks to our GitHub organization that it's well maintained by staff scientists in our group, and also data repository on OSF. And we always work with this principle of version control, but also code review. So each of us is assigned a buddy in the group or through our close network of collaborators that will also be the reviewer of the code. And we go into these cycles of iteration until we are convinced that the code is strong and it's tackling all the cases. On the other side, in the wet lab, we are starting to develop the approaches that we need for the experimental validation of each of these mechanistic indicators, and we are especially focusing on protein-protein interaction at the moment, where we can go from cellular assay that are based on pull down or co-immunoprecipitation, but then we can also go quantitatively with biophysical measurement. This is an example of an assay done to estimate KD that you can then try to correlate with the free energy changes. And we are starting to establish protocol for multiplex assay for protein stability, where we are starting with a small library for now, but then we want to scale up and using the sequencing here to have it as high throughput as we can. We cannot have the ambition to validate everything we generate, so we also try to reach out to groups that are expert in a certain, for example, cancer hallmark field or a certain protein target to see if they have data or they're willing to collaborate with us to get data on some variants that we predict interesting. And then we also reuse data available from the literature that are related to experimental data. And this generally for us, it goes using reanalyzing data that are deposited in databases with this deep mutational scan, and also literature mining when the protein is a little bit less under investigated. We still believe that there is a lot from publication we can use to inform on an effect of a variant that so we also maintain this part quite actively. And this combination of prediction and experiments started to populate our resource that it's MEBISP and has been designed as a conceptual and bioinformatic framework, computational framework with many tools into play. It's modular, meaning the way we design this, it's that you can also use the single building blocks. You don't necessarily need to go through all the steps for it, just what makes sense for a specific project. But of course, we have some, at least for when we release the data in the database, we have some requirements that need to be fulfilled of a basic set of analysis that need to be available on the target protein. And this scheme, it's just to convey the message that some of the modules are not really with the scope of giving us the mechanistic indicator, but more module to select and prepare the structure and quality control on the structure before giving the structure to the methods that do the prediction of the effect. And then the blue one are the one on the different mechanistic indicator, where within each of them, there is not necessarily just one method, but a combination of them. And sometimes we rely on a consensus approach to make the final classification. The minimal set of data for importing to the database, it would be to have at least assessment on stability and PTM, because for the way it is designed, they go together for some of the variants. We do this in two modes. One that it's really high throughput because we use just one single structure as an input, and then some of these methods have a little bit of conformational flexibility into account, but it still can scale up quite well. But then there is also more low throughput mode in which we dig more into the effect maybe of a subset of variants or a specific protein. And these rely a lot on ensemble from a classical simulation, from enhanced sampling approaches, or also experimentally derived ensemble. How this works in practice? I hope this scheme can help a little bit how we started to structure this within our group, and we presented it with the attempt of a living preprint. So if you go to bioRxiv, you see there are different versions of it, and it's building up the history. And then now we feel we have something meaningful, so we are starting also to submit the work for peer review. In the beginning, we did it for ourselves in a way, and then to have a pilot, and then we started to see how this can be used by other people. At the moment, the MEBISP team is going beyond my group already, because he's having people contributing from other research institutions. So we are moving more and more toward the community-driven effort, and we serve as the maintainer of the database and the collection point for the results. We can identify three main roles when we work into this. There are some of us that work as data manager, meaning they are the one with a lot of expertise already on this technique and resources, but they have also a good expertise in terms of as data scientist and sys administrator. And we are the one that especially serve in this final part of the process. I will get to there in 1 second. Then we are in contact with a group of people that have more an interest in developing method or workflow, either from our group or from other group, where we help them to understand what are the criteria of inclusion in the framework for a new tool or for the revision of an existing tool. And we work together until first we test it in some pilot project now before moving it to the official framework. But there are also people that have more of a bio angle to this because they are interested actually in doing protein analysis. Maybe they have a group of protein of interest for their own research that it's not in the database or it's missing some mechanistic indicator. They need to have a knowledge of protein structure and a knowledge of modeling, of course, so sort of expert-based work. But we help them providing the training on the tool and how to work with this. And at the moment, the training we provided was always based on a scientific research visit for short period of time to our group and some follow-up in remote. But we are also starting from the fall this year, we will start to launch regular training event, at least virtually online. And then we are on the way to organize one event every year physically at Copenhagen. So if anyone that it's in this network of students is interested in this for his or her own research, just reach out. The way we work then it's that the entrance point is a protein, and then the mutation are actually calculated saturation, meaning that we predict in advance all the possible effect for all the substitution. But then we have a way, especially through CancerMut and some other tool, to track annotation on the fact that this variant was already found in a disease, which resource is giving the information, so that we can recognize immediately which one are known variants from other clinical study and which one are potentially new variants. And this has been a big switch in the way we work, because in the beginning we were only taking the known variants. But then the clinician we work with, they asked, for me, this resource will be much more meaningful if I have all the prediction beforehand, because maybe I found a new variant in a patient that was never seen before, and I want to know if it can have some functional impact. So now we switched the saturation. So the entrance point, it's basically the protein of interest. Then the biocurator work mostly here with the tools to collect the analysis that are useful and to interpret the data out of this analysis, because we do also some reporting of the data and some visual support to the data interpretation. Whereas the developer are more maybe looking from the point of view of what we miss in the toolkit that can help us to understand more broadly impact of variation. Then we, as data manager, work a little bit more here, in which we the curator, they work in batches, so they are assigned batches of protein to follow. Then we aggregate the data. We have a step of data processing and validation to be sure that these data are as expected. The tool have been working as expected. Otherwise, we get back to the biocurator with some suggestion of where to focus for improvement. And then we have a local version of the database that we update every second week for all the people into the MAVISP consortium to use. For the public version, we want to spend more time on the quality control. We also have here a system of peer review to each other for the entries. And then we tend, for now, the capacity we have allow us to update the public's version every third month. The next release should be in the beginning of April, for example. Doing this, we have profiled already 600 protein. and a million of mutation. We gave priority to cancer tumor suppressor and oncogene, so you will see a bias maybe toward those in the database at the moment. This is to just show a little bit how we are growing over time, and it's a snapshot like before summer and during the fall last year. And when we have this remarkable increase here has been when we actually, for two reasons, because we switched to the saturation mode that allow us to profile more variants, and also because we ended up here finally with having a very good workflow based on SnakeMake or Dask and using and tuned to HPC computing so that we can really scale up with the protein we handle. In terms of data set that are already undergoing processing with some of the curator and using a sort of queue, we have a queue with priority assigned to it. It's covering, as I said, cosmic tumor suppressor and oncogene, a little bit more broadly ClinVar gene, starting from the one that are more highly mutated. We have a project in which we are focusing on transcription factor in their target gene. And then, of course, more broadly, protein involved in cancer hallmarks of specific pathway. But we also work on the benchmarking side. So we are always trying to find the resources of good data that we can use for benchmarking of the different models so that we don't rely only on what has been published by people testing those models. Can we also keep improving them and have an assessment over time? And then we have started a project that I hope to see more of this in collaboration with clinicians, where we get the data from the hospital on set. This is a cohort of childhood cancer, especially acute lymphoblastic leukemia, and we use the toolkit to profile the variants and add extra layer of information to the one you would have from the current approaches they use. I don't think I have very much the time to dig into all the aspects. So I thought I can just show you how we work behind the scene with the local interaction, because it gives a feeling also of other, for other models we reason and we work in the same way. In this case, the first step in the structure selection is the model that we call Interactome that has the purpose, and there is a toolkit for it that we are still developing, so you are free to use it if you need it for other things, but it's not fully constructed yet. In which we want to use the knowledge available in protein-protein interaction databases or available by experiments, so we still have a support for manually annotated interaction as a source of protein target interaction. Then for this, we link to the PDB to get to know which one are the complexes already with structure available, and also to curated resources with the AlphaFold based model that have been experimentally validated. So they are not just only generated by AlphaFold. For example, this study, we used model from this study from Beltrao group, in which they use cross-linking experiments to validate the model. And this allow, so the toolkit allow in the end to have an aggregated meta table with the ranked interaction from the one that we could trust more are a physical interaction from the point of view of what we know experimentally, and then also which one are available in PDB or not. And it's relying a lot on this PDB miner tool that Christine developed in the beginning of her PhD, that also can be a tool used in any case, even if you're not interested in a mutation, just to support you in your search of the best structure for a simulation or for modeling, because it's annotating and ranking structure from the PDB. When the structure in PDB is not available or we have a lot of missing residue, we rely on AlphaFold, and our protocol is a bit, now it's with AlphaFold 3 to build the complex, or also AlphaFold 2 and AlphaFold multimer. We still try to use a little bit of that. And then we need to pass some quality assessment with some score that are local or global on the complex before really giving this to the local interaction model for the binding free energy. In low throughput study, we also try to combine this with experimental validation before we really use the model ourselves. And then the local interaction itself, at the core of this, there is the concept of binding free energy calculation. And we use two different methodology, and we have designed a workflow to automatize this saturation and high throughput. One is based on the fold-X energy function, and our workflow is mutate-X, and the other one is based on Rosetta with this Rosetta DDG prediction workflow. The idea is that they come with their own limitation, especially fold-X has a problem of backbone stiffness because we don't move the backbone during the sampling. And Rosetta has a little bit of more local conformational changes that are accounted. So we try to use a consensus approach before classifying the variants, and we classify as uncertain for interaction if the two methods do not agree. And generally, this is a good example of variants to bring to the ensemble mode to get to see if the dynamics can rescue a signature. In doing this, we are starting to populate the MEVIS pre-interactome. So we can see that there are now a lot of complexes. Some of them are also very much interconnected. Probably if we plot this on a pathway, some of them will be in the same pathway, and meaning that we can start also to use our results in a more holistic way. And we started also to very recently to include the support for protein DNA interaction using this binding free energy approach. I wanted then to move to an example of the final outcome and how you can use the data also for your own interpretation. This is the example of PTEN. PTEN is a tumor suppressor gene. It was also a good example for us to benchmark a little bit our stability model, because there are deep mutational scan experimentally carried out with a readout on stability. So we could really see the accuracy in our prediction with our consensus approach for now. And we use this and other cases like this as a baseline now to see if there is a new tool that I want to test for replacing the current toolkit can actually increase our accuracy in the prediction, sensitivity and specificity, or it doesn't change the results. So we use it also to pass the criteria of inclusion of a new methodology. Then from the biological point of view, the results, this is the visual support. So there are aggregated CSV file with the numbers in it if you need to do more data-driven analysis, but also from the visual point of view, these help a lot to rationalize the data. So I will walk you through this dot plot because it's quite complex. So the idea is that we divided in blocks those features that are more annotation, either on the gain and loss of function or on the pathogenicity here, and also if they come with if we have experimental data. Then in blue are those mechanistic indicators that are more related to the protein functionality. So for example, long-range effect on protein interfaces, effect related to the PTM interplay with the mutation, or interaction between. This is an example. You could expand it and have multiple interaction here. Then the purple one are the ones that tell more about the stability and the interplay of PTM and mutation from the point of view of stability, that are this one. So and then the color code on the mutation side, if the variant it's already known in ClinVar to be pathogenic, benign, or if it's in this group of conflicting and uncertain variant. The way one could use this for application, it's, for example, start from here. Let's say that now these are all VUS and not pathogenic one. Start from here and filter out the ones that are likely pathogenic. Can I then see if they are loss or gain of function? And can I then associate to them a specific mechanistic indicator? But it can also be in a case like this, okay, they are pathogenic, we don't know very much on the mechanism, can I associate a mechanism to them, and can I experimentally validate the mechanism? We are already applying this, another group are applying this to important research questions. So you have a quick overview of what was published so far using MEVSP data in different ways. Funny enough, we started with the cancer angle, but then the first paper we published was actually for a relation of a protein that it's involved in a rare genetic disease, and this lysosomal storage syndrome. Here, it's a good example of how we could use the entire framework in a comprehensive focus study, because the protein is a protein attached to the membrane. So we actually used the ensemble mode here and simulated the protein in the membrane with a composition guided by experiments of the compartment where the protein should work. And we also validated against a literature correction of experimental data. It was interesting to see here as a take-home message that some of the facts were pointing to a lot of these variants with clinical impact hitting the stability of the protein, and something that was underappreciated in the field for this disease. And that I think it's really exciting for future perspective, because one of the treatment for people with this disorder is enzyme replacement. So if we can point out variants before I add that have a problem with stability, enzyme replacement could be a nice strategy for that. Then we also applied it to other neurodegenerative diseases. Here it's also another angle that give an example of how you don't need really the entire workflow and the entire framework. But for example, for this protein, TRAP1, in this study, in collaboration with Philomeni Group, that is an expert of redox modification, we were actually interested in looking effect in stability that we have already, and then to develop a new toolkit to expand on the portfolio of PTM and start to look into this S-nitrosylation that is a redox modification of the protein. So there is still some follow-up from our side to finalize the workflow for this so that it can be used on other protein too, and formally included in MEVSP. We also had another study on TRAP1 that it's now, I think, in press already on cell death and disease. In which here the scope was a bit different. Andrea Rasola group is working a lot on this protein from the point of view of mutation in cancer. They found a lot of mutation potentially clinically relevant, but they wanted us to help to use the framework prioritizing the one to bring forward for experimental validation, with the angle on the stability again, but also on the complexity of this protein, that it's a multimeric assembly in looking at the effect of the variants on the homodimerization, for example, or the larger assembly. We are also working in direction to expand the scope, and we want to move also toward more the field of intrinsically disordered protein, because they are really important in many diseases. So we are now work on the concept of short linear motif and functional motif for protein-protein interaction into, and now we incorporate this into the framework. And then the most exciting study so far that was published on Nature at the beginning of this year, it's how we joined forces with our computational assessment and a group that designed a multiplex assay in a mouse model for BRCA2 to get some functional score on the effect of these variants. And I have a figure about this. Yeah. So BRCA2 is a tumor suppressor gene, is involved in DNA repair. We are focusing mostly on a part of the structure because it's a very large protein with also more challenging region to tackle. But the majority of the, let's say, the hot spot for clinically relevant variants are in this structural part. The collaborator that approached us because they wanted to use MEVSP together with their data, they developed a CRISPR-Cas9 based saturation genome editing with a mouse model. And we did the assessment with different MABISP layer, more with the focus on the stability, but also those interactions that are important in stabilizing the protein, like SEM1. And then, for example, with this, we have, it allowed me also to introduce some other tool we have in the framework for interpretation. This is just an example of those that we could, that were already known to be pathogenic, that experimentally we could see that for the majority of them we could, and we did this on a large scale. We did this on thousands of mutations, but now I picked up some example because for visual purposes. We could see that also experimentally we found that there is a functional impact. And then from the mechanistic point of view, what we can gain from our framework here is that we can disentangle between the one that are altering this interaction with STEM1 and the one that are altering the folding of the protein itself. When we do the assessment, we don't only have the dot plot, but we have some more compact way to look at the data. For example, to plot for the mutation the different mechanistic indicator. So you can also see the interplay between effects. And when it's maybe just one effect triggering the signature that are this lollyplot. And in this study on BRCA2, then in the end, we reached out with clinicians that had data from patient. And the combination of all the data allowed us to reclassify some variants that were uncertain into either benign or pathogenic and bring knowledge to the ClinVar database itself. In the future, what we are trying to do, and we started to do from the beginning of this year, is to have a support also for the effect of mutation on protein-drug interaction that we don't cover yet, and still using this free energy calculation concept. So we rely for now on a alchemical free energy protocol in the ensemble mode. And we are scrutinizing for inclusion other tools that are maybe more machine learning based and could fit into the simple mode, but we are not convinced yet. And it's also a field where we would like to collaborate more with experts because it starts to be out of our comfort zone. We have for a project the need of moving further from missense mutation to also insertion and deletion. So we are looking into this. As I said, we are expanding the PTM support and both with the redox modification and starting in the summer this year, we will start with glycosylation because of a network of PhD student supported by Marie Curie grant that will focus on this topic. We look always for inclusion of additional method or new sampling approaches, and expression of interest for people that want to be credited as developer will be very welcome. And I think we need to join efforts if we want this to become a meaningful resource for everyone. We do training activity for biocurators, and also here expression of interest are very welcome. And then the biocurator get credited for their work. They get also included in the publication when we introduce the new version of the framework and the new data, or if their data are used for research. We also want to try to reuse available data that are demanding from the computing point of view. So there is always a look into the possibility of reusing molecular dynamic trajectory that satisfy our inclusion criteria, because we have certain requirement on force field and simulation length and so on, or model that have been experimentally validated for protein-protein complexes, or that satisfy the quality control that I presented before. And then I want to close thanking all the people in my group that over the year made this possible with all the ideas, creativity, and motivation that they put into it. The people that are also contributing from other institutions and that started to make MABISP something that goes beyond our group and something that can be meaningful to the community. Especially I would like to thank Burcu, because she started actually here, and we did the prototype of this, but now she's still collaborating from outside. And then our collaborators that are interested in this data because they either have clinical data, some of them, they do experiments, they have methodology that are of interest. And thank you for your attention.
Thank you for the precious talk, dear Dr. Papaleo. We can continue with the question and answer session. So you can write your questions to the chat box. It is especially written as question and answer. So
Or we can also take, like, if you can just unmute yourself and ask your questions.
Yeah, I think it will be nicer and maybe
Yes,
more
yes.
interactive like that. Yeah.
Should I start maybe then? Or anybody? Okay, it's a fascinating presentation and fascinating, like it's very interesting, and I think it will be huge effect in the cancer biology, and also people who are, the main, I think anybody who does protein dynamics or studying something computational biology, eventually you need to find a stabilization, something that stabilizes your protein. And I think maybe it's a little bit, off the topic, but I think it also will affect somehow that kind of research. I don't know if I'm pronouncing right. MAVISP, right? The frame. Okay, great. So maybe my question will be a little bit naive, but I know I know you have many different like the modes, but my question is basic for saturation, for like, trying to find stabilizing variants. So you are using 2-3 different softwares, I guess. One of them is Foldex base.
Yeah, so at the moment, so we look if the variance is predicted to be either stabilizing or destabilizing, because then in disease, especially with tumor suppressor, that it's an interesting signature. At the moment, we have included Foldex, Rosetta-based approach, and also RASP, that is a machine learning model that has been developed on top of Rosetta. So RASP was trained on Rosetta data, and this is because Rosetta is really computationally demanding. So if you want to have a good assessment on this effect on stability, high throughput as we have here, indeed it helped us a lot in terms of speeding up the process and of data acquisition, and it has still a good accuracy. We are keeping in parallel a benchmarking study using protein that have not been, because we produce the data on new protein. So now we can actually benchmark RASP against the others, because we can take the protein that have not been used in the training and still the accuracy hold, and it's really promising what we are seeing. We are also interested, we always keep an eye on other methodology that we could include here, where, as I said, now to be convinced that we should include them instead of Foldex or Rosetta. We want to see that they can improve the accuracy of the prediction compared to the experimental deep computational scan data. That it's ultimately what we are interested in, because we are using this to say what could be the effect that I measure experimentally. And then the other thing that could be a criteria of inclusion there for new methods is also considering the computing resources and the time that you need. That was one of the criteria with RASP. We wanted to see that it was still accurate enough for our purpose, and then we have to gain from it compared to Rosetta. So we are also looking into other approaches. There is one thing to consider here, that when we want to predict stabilizing mutation, that it's where you were mentioning, there is a lot of bias in many of these methods. Because they are more, it comes also from the history, because we have much more data on destabilizing mutation. So there is a bit of bias in that direction, and in that sense, it's really difficult for us at the moment to get a clear signature of mutation that could stabilize the protein. Sometimes we have them, and then it's where we prioritize the experimental validation, because we don't necessarily trust that. Yeah,
Last time I checked, I think the accuracy for any kind of software for predicting a stabilization was like around 20%.
yeah,
But
it's very
yeah,
yeah.
I know it's very bad. But you have also the experimental data, and then as you said, I think maybe this is a little bit off the topic, but how many researchers are working on this? It's a very huge project,
it's
demanding,
very
right?
huge project. It goes on and off in the sense that, for example, for the people doing the biocuration, it's not that they are active all the time. They tell when they have time to work on this. So we have some period of time in which we have more protein to process than other. But as an average, at the moment, we are 20 people actively working on this. And I don't know, if we grow, we can do more. But
Like, can we volunteer to do something, or you are?
yes, yeah, that's what we want to try to do with these training events, because then we also need to see, it can also be a good opportunity for students. For example, if you have some protein that you are studying for your own research and they are not in the database, then you can become the curator for those. And then you also gain that the data are deposited and maintained there. Yeah, so we do that. And what I suggest is to actually write me, because then I can see when I have maybe a number of expression of interest, we can organize a first training event online. Some of the students in the past that approached us, they were then applying also for some fellowship for short-term visit, because they felt that they wanted some interaction in person. So they stayed like 1 month with us. We did a full immersion, and then they were working in remote.
Okay, great.
Yeah, so it's possible, yes. So we just need to figure it out the way. And at the moment, we also give access to the computing resources if that it's becoming an issue.
Oh, I see. Particularly for the AMD simulations, I think it will require both the computational power and the storage, I guess, right? So you also need huge storage data.
Yeah, I see. So we have a local storage. Then we are trying to figure it out what it's the best way also to release the trajectory we generate for other people. And for now, it's very protein centered. So if you go to OSF, you start to see a repository for each protein. But I'm also trying to follow the work of some initiative for molecular dynamics databases, because we would like eventually to deposit the data there. Another thing that in terms of collaboration, it can also be if some researcher, they have their own molecular dynamic simulation. And it's a protein that we are already having in the simple mode. It would be really nice to use that trajectory for reanalysis for us, so we don't have to generate the data again. There are some criteria of inclusion there that it's for us, we are using charm 36 as a force field. So we don't want to, just to keep some data, if we want to use it in benchmarking, that have been produced more or less the same way. So we don't want to change the force field at the moment. And yeah, that I think would be the main bottleneck. And then the simulation should be at least 500 nanoseconds for the properties we are assessing now. We are doing some study to see if we can go shorter, but we are not there yet. Yeah.
I take a lot of time, but I mean, if anybody has a question, they can just unmute themselves, or I can continuously ask questions. You also consider illusory, right?
Yeah.
Okay. Like cross correlations, or you have any other?
No, so in the simple mode, it's an elastic network-based model. It's AlloSigma. I don't know if you're familiar with it. For us, at our, I mean, maybe we are missing something in the literature, I don't know, but for us, even from our recent literature search, it's still one of the few approaches that really managed to develop a score that is based on free energy concept for mutation, for allosteric mutation. So we like it a lot as a first screening. And then in the ensemble mode, well, you know, Pinterest, I guess, and our protein structure nectar approaches. So we are still using that, and we are starting to include also the dynamic correlation, but we are not there yet. So
I
it's
see.
more topology based, I would say.
I see. And this is last, I promise. Disordered proteins, have you ever looked anything for the disordered proteins, or it is completely out of the...
We started, we don't have many proteins. Like that. So for the local interaction, we can already do a lot, because if you use this principle of short linear motif, then you can still use the free energy calculation scheme. The problem is to have a good model of the interaction between the SLIM and the protein. But also AlphaFold based method are becoming quite successful with that. So local interaction, we can do it, no restriction there. Stability, I don't think it matters that much to talk about stability on an IDP from the point of view of the stability of the structural assembly, because but there are other properties you might want to account for example, how the mutation is changing the compactness or the secondary structure propensity, because those are generally important in the molecular recognition. So we are working in adding new modules and new indicators that are IDP specific. Yeah, we are working on that.
I was asking because for a normal disorder like flanking regions we cannot do anything, but multiple, like many of the disordered regions have like one, maybe like a short sequence which is structured, and then a flanking regions. So some of the research right now like trying to target how you can change that structural motif. This is what I was trying to ask, but I totally understand it's very...
Yeah, but that so if we look from the point of view of the structural motif, yes, we can work with that. Yeah,
I see.
We were still wondering. Yeah, that's an interesting point, because we were still wondering in the group if, for example, we should still include the IDP region from intrinsically disordered protein that are maybe a contiguous helix in the stability model, or not. So at the moment we are not putting them there, but it could be information that we miss in the end.
I see. Very, very fascinating. Thank you so much. We can take any questions.
I don't buy it.
Because I ask everything. This is why. Okay, then thank you so much. Like I really,
Thank you for your time and invitation.
Yeah, thank you so much, and we are also thanking all of the participants.
Yeah, thank you.
Thank you.
A weekend when you come to that. Bye.
Bye bye.
Very nice.
Bye bye.